Hey guys, today I wanted to talk about the physics of action potentials. Just a quick reminder before I get into the biology of action potentials. Two very important concepts we covered earlier in the semester were Ohm's law and capacitance. Ohm's law is V equals IR, which relates the current and resistance to the voltage. And capacitance is equal to the permittivity of free space times the area of each plate divided by the distance between the plates. And capacitance is the ability of the system to store an electric charge. I know most of y'all are bio majors, so I'm sure you're familiar with a neuron, but just in case you aren't, or if you need a reminder, a neuron is an electrically excitable cell that communicates with other cells via specialized connections called synapses. They're mostly found in the central nervous system, which includes the brain and the spinal cord. And as you can see, we have a picture of a neuron here, and the action potential is generated in the axon hillock and travels along across the axon into the synaptic terminals, where it then communicates with other cells via the synapses. Along the neuron, there are many voltage-gated ion channels, which are very important because they are the main driving forces of the action potential. And sodium, potassium, calcium, and chlorine are major ions that are responsible for the changes in the membrane potentials. The intracellular and the extracellular solutions in the body are ionic solutions, and therefore will conduct electricity. And these ions can cross these membranes through ion channels that act as resistance in an electrical circuit. And a cell's resistance is the measure of how easily the ions can move through the membrane. And when a cell is at rest, most of these ion channels are closed, and the cell is described as being in a high resistance state. A cell's capacitance determines how quickly the membrane potential can respond to a change in current. As we learned in class, a capacitor is made up of two conducting materials separated by an insulator, and the extracellular and intracellular fluids are the conductors, and the lipid membrane is the insulator. And the membrane capacitance is related to the size of the cell. So therefore, the larger the cell, the larger the area, and the more lipid membrane there is, and therefore the larger the cell's capacitance will be. When there's a voltage difference, such as the resting potential across the membrane, charge will build up at the interface because current can't flow directly across the insulator. And when this built up charge becomes large enough, an induced current is produced, which will change the membrane voltage. Sufficient current is required to initiate a voltage response in a cell membrane. And if the current is insufficient to depolarize the membrane to the threshold level of negative 55 millivolts, then an action potential will not fire. But if it does reach that threshold, then the membrane will depolarize as sodium enters into the cell to about positive 35 millivolts. And after that, it will repolarize as potassium flows out of the cell and will actually dip below the resting membrane potential. And this is known as hyperpolarization because the potassium channels close slowly, and then there's a brief refractory period before it returns to that resting membrane potential. Many neurons contain a substance known as myelin, which is an electrical insulator that speeds up the action potential. And it does this by decreasing the distance between the conducting materials, which then in turn decreases the capacitance. And it also increases the electrical resistance, which prevents the current from leaving the axon. And the myelin assembles voltage-gated sodium channel clusters at these discrete nodes along its length, known as nodes of Ranvier. The sodium channels are only at these nodes, and so when one sodium channel opening depolarizes a much greater length of the axon until it reaches the next node of Ranvier, where the voltage gate sodium channels open and the cycle continues. So essentially, these action potentials hop around, covering much greater distance in a shorter amount of time. This is known as saltatory conduction, which is the propagation of action potentials along myelinated axons from one node of Ranvier to the next. And as it travels on the axon, it finally reaches the synaptic terminal, and then depolarizes the membrane, and the voltage-gated calcium channels open and calcium flows in, which triggers the synaptic vesicles to release the neurotransmitter, which then binds to the receptors and communicates with other cells in the body. And that is it. And here are my sources. And thank you guys for listening.